plant breeding for disease resistance. Here we can incorporate a different desirable qualities into a single variety. Here if the plant is having high yielding quality, but without resistance, so it becomes wasteful. So if it is giving more yielding, but at the last stage if it is affected by different diseases, so that is very great loss to the farmer. So that's why here we can improve the disease resistance, here plant breeding for disease resistance. So here every year in the total crop 20 to 30% of crop is loss, that is by here different pathogens or diseases. 20 to 30% of crop loss by different pathogens or diseases. So sometimes the total crop also, so it is affected by the disease or pathogens. So that's why we can improve the quality or disease resistance. So here the disease resistance quality is incorporated into a high yielding variety. So then they are giving the high yielding and also they are showing the disease resistance. So here the we can improve the disease resistance by here two methods. It is by a conventional method and by a mutation breeding. By conventional method or traditional method, so in the germplasm, so only a few or limited genes are present. So if the germplasm is having very limited genes, so we cannot incorporate the disease resistance quality. If it is present in the germplasm, so we can incorporate the quality into the hybrid variety. But here in the by conventional method, so it is the by conventional method we can produce only a few genes or a few varieties. In the by conventional method, here the germplasm is having only a few genes. That's why here if the limited genes are present in the germplasm, so it is not possible to incorporate all the qualities. So then we are shifting to the mutational breeding. So here in the conventional breeding method, here only a few limited genes are present. So it is not possible to incorporate all the qualities. That's why here we are using a mutational breeding technique also. Here what is mutation? Here mutation is the sudden inheritable change. So it is the change is passing from one generation to the next generation. So that is called as mutation or change. Here the mutation is also, it is genetically the genetical change. So what is genetical change? So in the change in the base sequence, change in the base sequences within the DNA or within the genetic material. The genetic material, it may be DNA or RNA. Here the change in the base sequences. So bases means nitrogen bases. In the DNA is composed of number of nitrogen bases or nucleotides. So if one nucleotide is changed, that is leading to the great change or great morphological or physiological change. So change in the base sequences. So here one nucleotide change also leads to the great morphological or physiological change in the body of organism. That's why here the mutational breeding. So how the mutations are taking place in the organisms? The mutations are induced by different mutagens. The mutations are caused by different mutagens. So which is called the material or the material which is causing the mutation, it is called as mutagen. So the mutagens are physical mutagens are there and chemical mutagens are there. Here mutagens like mutagens here physical and chemical mutagens. So, physical mutagens like X-rays, gamma rays and beta rays. So, different physical rays are there. So, the ultraviolet rays, they are inducing the mutations or they are changing the base sequences or nucleotide sequences. So, they are leading to the change in the nucleotide sequence in the DNA or genetic material. So, physical mutagens like ultraviolet rays, X-rays and chemical mutagens. Here chemical mutagens like nitrogen mustard gas or ethyl methane sulfonate, methyl methane sulfonate and sometimes colchicine. So, these are also changing the base sequences or sometimes the number of chromosomes also increased or decreased. So, the physical chemical mutagens are causing or inducing the mutations or changes within the genetic material. So, that is leading to the morphological or physiological change in the organism. If these changes are useful to your organism or for commercial purpose, if the changes are useful, so we are using the type of mutations. So, here some plants are induced by chemical or physical mutagens, then the plants, if the plants are showing the disease resistance, before the induction the plants are not showing the disease resistance quality or trait, but after the induction of mutations, after mutating, the plants are showing the disease resistance. So, then the plants are useful or commercially important. So, that is useful to the farmer. So, here number of plants are here induced by here mutagens and one example for here mutation, here bindi or lady's finger, okra, it is also called as okra. Scientific name is Abel muscus esculentus. Abel muscus esculentus. So, it is after the induction, here the bindi plant is showing the disease resistance. So, here it is generally the bindi plant is it is susceptible to yellow mosaic virus. Here the after the induction, after mutations, after the induction of mutations, here this plant, bindi plant is here it is showing the yellow mosaic, it is showing the disease resistance to yellow mosaic virus. So, here the some plants, some wild varieties are having the disease resistance quality and some plants are giving the more yielding, but easily susceptible to diseases. So, the wild plants generally, wild varieties are having the disease resistance quality and with poor yielding or low yielding, and some other plants are having the high yielding capacity, but no disease resistance. So, that's why here that these two plants, wild variety with disease resistance and other variety with high yielding quality. So, the two plants are crossed or the here the disease resistance quality is incorporated into the high yielding variety, or these two plants are crossed. When these two plants are crossed, the hybrid variety is showing the high yielding quality and disease resistance. So, like this here by plant breeding technique we can incorporate the disease resistance quality, and by mutational breeding technique also we can incorporate the disease resistance quality. Like this, by mutational breeding, here one plant is developed, bindi plant, Abel muscus esculentus, lady's finger or okra. So, it is showing the yellow mosaic, it is showing the disease resistance to yellow mosaic virus. And some other plants are also here developed, here the wheat, brassica and cauliflower, cowpea, chili plant. All these plants are here generally they are susceptible to different diseases. Here the wheat plant generally here it is susceptible to this leaf and stripe rust and hill bunt. So, after here after the incorporation of this quality, disease resistance quality, so it is showing the here it is showing the resistance to leaf and stripe rust and hill bunt. So, this variety is called as Himgiri. Himgiri, the variety name, here it is the crop, here variety or a hybrid variety. And it is showing the disease resistance to the plant. This variety is showing the disease resistance to leaf and stripe rust and hill bunt. Brassica, here mustard plants are also here. Mustard plant varieties Pusa, Swarnim or Karanrai. So, this plant is showing the resistance to white rust. And cauliflower, here it is also Brassicaceae family member. Here it is Pusa, Shubra, Pusa, Snowball, K1. So, these are the varieties or hybrid varieties. Pusa Shubra is showing the here disease resistance to black rot. Pusa Snowball K1, it is resistance to cull blight, black rot. And cowpea, here Vigna plant, Vigna, scientific name is here Vigna. Vigna anguiculata, here cowpea plant, here the variety is Pusa Komal, showing the resistance to bacterial blight. And chili, here the chili is here variety is Pusa Sadabahar, and it is showing the resistance to chili mosaic virus, tobacco mosaic virus and leaf curl. So, the pathogens here, the disease is by different pathogens. The pathogens are fungal pathogens are there, here bacterial pathogens and viral pathogens. So, generally the diseases are by fungi, bacteria and viruses. Here the fungi generally these are causing a number of diseases by different pathogens, fungal, bacterial and viral pathogens. Here fungal pathogens like Phytophthora infestans, and Puccinia recandita, Coletotrichum falcatum, here Phytophthora infestans, Puccinia recandita, Coletotrichum falcatum. So, these are some fungal pathogens causing here different diseases and bacterial pathogens. Here bacterial pathogens like Xanthomonas, Campestris, Oxonopodis citri, variety citri. Here viral pathogens, here tobacco mosaic virus, cauliflower mosaic virus. So, these are the some viruses. So, different pathogens here fungal, bacterial and viral pathogens are causing the diseases. So, here we are improving the plants with here disease resistance quality. So, here the plants are showing, some plants are showing the here these are the pathogens, fungal pathogens, bacterial pathogens and viral pathogens. They are causing number of viruses, bacteria and fungal pathogens are causing disease. So, here the plants are showing the disease resistance. When the disease resistance quality is incorporated into the variety or into the plant, these plants are showing the resistance to different diseases. So, here the disease resistance we can incorporate into the plant by plant breeding technique or by conventional method or by plant breeding techniques or by genetic new techniques like genetical engineering or molecular techniques. By using molecular tools also we can incorporate all these qualities, or disease, not only disease resistance, here different qualities also we can incorporate. So, here after the incorporation of disease resistance quality or gene, so that is showing the disease resistance. So, it is about disease resistance here developed by plant breeding technique.